Now, so far we have discussed sterilization. Now, let's talk about endotoxin inactivation. What is this endotoxin? Why are the regulators so concerned at every stage of sterile processing about the presence of endotoxins? What are they? It starts with pyrogens. Pyrogens are those that induce fever-like conditions in the body. They could be chemical substances, they could be biological substances. Bacterial endotoxin is one such, what we call exogenous pyrogen. Now, it doesn't come from all bacteria. It comes from gram-negative bacteria because they have a cell wall. And this endotoxin is part and parcel of that cell wall. And like the human skin that sheds skin cells throughout the day, throughout the year, throughout your lifetime, this bacterial endotoxin on the membrane wall also flakes off every now and then, and it falls inside the body of the cell. The day the cell wall is broken or damaged and the contents come out, those are the endotoxins that come out. And they serve as exogenous pyrogens. What is the meaning of that? We should first understand how the bacterial endotoxin has any effect on the human being. Once it gets into the body, the bacterial endotoxins trigger a mechanism by which you have RNAs, the different RNAs, messenger RNAs, relay RNAs, transmission RNAs, whatever. They get into an act and they trigger the production of endogenous pyrogens in the body. The pyrogens in our own body are not kept in one place ready for use. They are manufactured on demand. And the exogenous pyrogen triggers the production of endogenous pyrogens in our body. And that sends a signal to the command center that body is losing heat, body is losing oxygen, so there is tremendous increase in the oxygen consumption and a tendency to try and control the body temperature. And that's why you start having elevated body temperatures. Now, this is a phenomena that all of you are familiar with, that endotoxins means fever. If it was so simple, no regulator would have lost so much sleep. This bacterial endotoxin has other pathological aberrations that it initiates in the human body. It compromises and alters the carbohydrate metabolism, the lipid metabolism, and the hemodynamics in the body. For example, it triggers the platelets into releasing thrombin and fibrinogen and causes the blood to start clotting. Once the blood starts clotting in the body, you go into a state of severe hypotension and you go into something called a shock, hemophilic shock, and that can be fatal. It is this other phenomena, amongst other pathological aberrations, that keep the regulators very concerned and focused on the control of endotoxins. The gel clot test that you people use, the gel clot assay, is a manifestation of the same effect, the clotting of the blood in the horseshoe crab, the LAL test. Now, what is this endotoxin? In the microorganism, if you were to take it out, you will find a mixture of lipopolysaccharide, specific to that particular microorganism, carbohydrates, proteins, and so on and so forth. Now you purify this lipopolysaccharide and you have purified endotoxin. Purified endotoxin has a far more devastating effect than the normal mix that comes out of a microorganism. Now what is this lipopolysaccharide? Lipo is fat, is the main component of the endotoxin, and it is this lipo element that induces the reaction in our body. The immunoresponse is elicited by the lipo, and that is common for all gram-negative bacteria. Now to the base lipo is the polysaccharides, the sugar chains. You have the core oligosaccharide, and then you have the O-specific chains that could be several repeater units, 20 to 40 repeater units, each repeater unit having 3 to 5 different sugars in a particular sequence. And it is this that characterizes one microorganism from another. The polysaccharide element varies from species to species of gram-negative microorganisms, but the lipo part remains more or less the same. And that is what induces this immunoresponse. So what we can do are two things. One, eliminate, remove the endotoxin, or inactivate the endotoxin in such a way that it does not anymore elicit any immunoresponse from the body. Now, how do we eliminate bacterial endotoxins? Activated charcoal, reverse osmosis, ultrafiltration, affinity chromatography, alkali wash, sodium hydroxide, potassium hydroxide. In fact, those days when LVP bottles were used, LVP was supplied in bottles. One of the standard operating procedures used to be, after washing the bottle, is to give it an alkali rinse and then wash it once again with water for injection and then take it up for production. So alkali chemically combines with this lipopolysaccharide and takes it away. In the case of thermal energy used, we can only inactivate the endotoxin. So there what we do is to try and change the spatial configuration of the lipo part of the endotoxin such that it is rendered non-immunoresponse eliciting type. In other words, you change the configuration such that it is no longer antigenic. Now, how much endotoxin can the body take? It has been found that it is 2 nanograms per kilogram of body weight. Now, the release specifications for formulations is not more than 0.5 endotoxin units per ml. In the case of water for injection, it is 0.25 endotoxin units per ml. And how much is this? 1 endotoxin unit is equal to 0.2 nanograms of endotoxin, purified endotoxin. And you get them in vials. Each vial has got 10 raised to 4 endotoxin units. Now, that is called the reference standard endotoxin, based on which you could have a secondary working endotoxin standard, that is a control standard endotoxin. The US FDA asks you for 3 log reduction of E. coli purified endotoxin. The European Pharmacopoeia tells you 250 degrees centigrade for 30 minutes or 200 degrees centigrade for 60 minutes. And finally, there's something called endotoxin recovery and endotoxin that is residual. Endotoxin recovery refers to spiking an item with a known amount of endotoxin and being able to recover it while it is still untreated. Whereas endotoxin residue relates to the amount of endotoxin remaining after the item has been exposed to inactivation treatment. So just as in cleaning validation, we take swabs and then carry out studies, recovery studies to see how much did we spike and how much is being recovered and what is that percentage. The same study needs to be carried out for endotoxin recovery as well as for endotoxin residue determination. Now you see, endotoxin, bacterial endotoxin has got different levels of affinity for different substrates. For some polymeric surfaces it has got very high affinity, and for some polymeric surfaces it got very low. The recovery from some of those polymeric surfaces can be very consistent and very high, and in other polymeric surfaces it could be very low but consistent. In the case of borosilicate, you find that the variability is over a very wide range in terms of the endotoxin recovery. But whatever it is your job to carry out the study and establish what is the mean recovery percentage that you are having for both the endotoxin recovery study as well as for the endotoxin residue determination after inactivation.